Hi, I'm Art Craig, and this is a talk I gave at a vaccines research and development workshop on 11th of November 2024 on applications for mRNA therapeutics, the immunological issues and considerations. And of course, the central dogma of biology is DNA is transcribed into RNA, is translated into protein, and traditional drugs target proteins. In the case of mRNA therapeutics, we're bypassing that process by introducing an exogenous mRNA to be translated into a protein. And the applications where we may want to do that are to substitute for a defective gene, like a lysosomal storage disease. In that case, we do not want an immune response to the product of our mRNA therapeutic, or vaccine settings where we do want a response. And the most common vaccines are infectious disease vaccines, such as COVID, where we're especially interested in inducing protective antibodies, and cancer vaccines where we want to induce anti-tumor CD8 T cells, which is much harder, so I'll be focusing on that. Now, in both of those cases, when we're introducing exogenous mRNA, we have to overcome defenses that have evolved against these exogenous nucleic acids. And these go back into archaebacteria that had to protect themselves against bacteriophage that would hijack their DNA or their nucleic acids. This whole area has been termed nucleic acid immunity to refer collectively to the set of defenses that have evolved to protect genome integrity, starting again in archaebacteria. These include nucleases to degrade foreign nucleic acids, RNA interference, the CRISPR-Cas system, a variety of RNA and DNA modification enzymes, and the nucleic acid immune sensors, which I'll be talking about mostly in today's presentation, including toll-like receptors, RIG-I-like receptors, NOD-like receptors, and the cGAS sting pathway. Now, all of these mechanisms have been used in the development of nucleic acid therapeutics, but I want to just focus today on the importance of these nucleic acid immune sensors. These are a subset of the pattern recognition receptors at a high level. There are two important ones which target DNA, including TLR9 in the endosome, which distinguishes self from non-self based on unmethylated CpG dinucleotides, and cGAS sting in the cytoplasm. cGAS sting pathway does not distinguish self from non-self. It's activated by self-DNA in the nucleus or by exogenous nucleic acids. And then for the purpose of today talking about mRNA therapeutics, I'm going to focus on the RNA receptors. These each have some ability to distinguish self from non-self, which is very important in the case of therapeutics. And so coming back to the three classes of mRNA medicines that I mentioned, for protein replacement, we want to avoid all of these sensors. And that can be accomplished by substituting pseudouridine for uridine to avoid activating TLR7 and 8, excluding the double-stranded RNA byproducts of in vitro transcription reactions that can activate RIG-I-like receptors, and using an immune-neutral delivery, avoiding immune-stimulatory lipids, for example. mRNA vaccines to induce antibodies to prevent infections have really, of course, succeeded enormously in the case of COVID, and the solution there has been to substitute pseudouridine for uridine, again to avoid activating the TLRs that induce interferons and injection site reactions that can be problematic, and instead to include double-stranded RNA byproducts that provide an adjuvant effect by stimulating RIG-I-like receptors. Now, for mRNA cancer vaccines to induce anti-tumor CD8 T cells, there has been some initial success, partial success, but it's unclear how it may be possible to further improve those. And so that's the focus of my presentation today. Just to go back to basics, all vaccines start with the concept of signal 1, which is antigen, and signal 1 alone leads to anergy, does not induce an immune response. You need signal 1 and signal 2, the costimulatory signal through CD80 and CD28. And signal 2 is not constitutively expressed, but has to be induced. And that induction is where pattern recognition receptors come in, providing the initial signal that induces the expression of costimulation. Now, in addition to that, to get maximal CD8 T cell expansion, you also need a signal 3, and IFN-a, the type 1 interferon receptor, is the best understood of that. IFN-a is activated by all 18 of the human type 1 interferons, and this signal 3 is very important for maximizing the CD8 T cell expansion. CD4 T cells, to help B cell make antibodies, are less dependent on that co-stimulatory effect compared to the CD8 positive T cells. Now, 18 different type 1 interferons, these have very diverse immune effects that are not well understood generally, and so I really want to dive into that a little bit more. The key point at a high level is that interferon alpha drives CD8 positive T cells in humans, whereas interferon beta has a fundamentally different biologic effect driving Tregs. And this is not actually widely appreciated, despite the fact that when you think about FDA approvals for interferon alpha and beta, it's obvious. During the 1980s, when these first became available in recombinant form, interferon alpha and beta were used to treat just about every disease that people could imagine. And very early on, interferon alpha showed some activity in treating cancer. It was eventually approved for treating HCV and HBV. Whereas interferon beta, despite being tried in multiple different indications and clinical trials, did not succeed in gaining FDA approval in those indications. Where it was approved instead was in the treatment of multiple sclerosis, where the mechanism of action appears to be activating Treg cells, not the CD8 T cell responses that are involved in efficacy for treating cancer and chronic viral infections, and IL-10 induction. So how does this happen? Why do these interferons, which both work through IFN-a, have such different biologic effects in humans? The binding has a different affinity. Interferon beta has a much higher affinity than interferon alpha. So under conditions where both are present, interferon beta presumably will dominate. They bind the receptor in a slightly different way, but it's really not well understood why the downstream effects are so different. One other point, there's one interferon beta gene. There are 13 different interferon alpha genes. They're closely related, but all of these have been conserved through evolution. They are non-redundant, and we really don't know what's different about those genes that they've all been conserved so highly. Now, in mice, the system is different from in humans. In mice, interferon beta actually drives CD8 T cells and tumor responses better than interferon alpha. So mouse models, I mean, it's long been clear they don't predict human efficacy. This may be just one reason, but it makes it very difficult or even impossible to extrapolate from innate immune activators in mice to predict their results for humans. So if we're developing human vaccines, the proposal that I would make is that we really want to induce the highest possible ratio of interferon alpha to interferon beta if we want to achieve this type of effect rather than that type of effect. I just don't see how we can get around that. So then we have to turn to thinking about the cells that produce these different interferons. Interferon beta can be produced by essentially all nucleated cells in the body, and it's made in response to just about any pattern recognition receptor that's activated. Interferon alpha genes, on the other hand, are primarily transcribed and expressed at high levels only in this one subset of the plasmacytoid dendritic cell called P1 PDCs. This makes about more than 90% of the interferon alpha that's produced in response to infection. And there's only two receptors that trigger these P1 PDCs to make that strong response. Those are TLR7 and TLR9, which again, as a reminder, detect viral RNA through uridine and viral DNA through unmethylated CpG dinucleotides. So what are P1 PDCs? Most of the people working in cancer immunotherapy actually haven't heard of them before. These were defined in 2018 based on the staining for PD-L1 and CD80, where naive PDCs don't express either of those. And after 24 hours of flu virus expression, flu virus stimulation. P1 PDCs are one of the subsets that differentiate based on positive for LDL1, negative for CD8. And this is the subset that functionally makes interferon alpha and has been reported to defend against COVID viral infections early in the course of infection. The P3 subset, on the other hand, promote Th2-like responses, and it's been reported that P1 and P3 frequencies are inversely associated with melanoma survival, high P1 PDCs being good and high P3 PDCs being a negative prognostic factor. So let's just hypothesize for a moment that maybe P1 PDC induction via TLR7/9 might promote anti-tumor CD8 T cell responses. And in the same manuscript, the authors looked at a panel of different innate immune activators for their induction of P1, P2, and P3 PDCs. And I've just extracted that data into this table on the right, where I've ordered these different innate immune activators in order by the percentage of PDCs that differentiate into that P1 subset that makes the interferon alpha. And then I've also calculated the ratio of P1 to P3 PDCs. And what's striking here is that when you layer into this table the data from published clinical trials of different agents in humans in the treatment of cancer, the only agent that has shown systemic monotherapy activity in humans at more than 10% is a CpG-A DNA that also induces this highest P1 level. This is vidutolimod. Flu virus, of course, hasn't been used therapeutically in humans, but TVEC is the next closest thing. And then we have the different CpG oligos and TLR7/8 small molecule agonist R848. So only vidutolimod induces more than 10%. I just want to take a moment to talk about how this differs structurally from the other TLR9 agonists, because a very important point that I want to make is that the structure of TLR9 and TLR7 agonists plays a very major role in the immune effect that you get and the therapeutic efficacy. So CpG-A DNA up on top here, uniquely among these CpG DNAs, has a native phosphodiester DNA backbone, contains a palindrome that's underlined. This has a palindrome too, but it doesn't have a native backbone. It has a phosphorothioate backbone. And it forms G quadruplexes. These G-rich regions on the ends stack into G tetrads that assemble into G quadruplexes. And G quadruplexes are present in the genomes of all retroviruses and viruses. So really, the CpG-A DNA is mimicking the viral genomic structure much more than these phosphorothioate backbones. The phosphorothioate backbone still can be a very good drug. There are 2 approved vaccine adjuvants using CpG-B. It's a very strong B-cell activator, induces very high antibody responses. It just doesn't induce a lot of P1 PDCs. And in clinical trials in oncology, it really did not show very much monotherapy activity. Neither did the CpG-C. So now turning to RNA, what about mimicking viral RNA to activate TLR7/8? Because there's multiple different agonists for TLR7 and 8. We know from the crystal structure of TLR7, it forms a homodimer that contacts RNA at 2 sites. There's a first site that binds guanosine and a second site binding UUU or NUN. And that induces interferon alpha from the PDCs. It doesn't have a lot of inflammatory effect, very low IL-6 and TNF. Now, small molecule agonists for TLR7 bind very strongly at the first site, but they don't interact at the second site. And the consequence of this is there's an allosteric shift that results in activation of the NF-kappa B pathway downstream from MYD88. And that can easily be measured in NF-kappa B reporter cells that were used initially to characterize the biologic effects of that. Or in a human PBMC assay, you see very strong induction of IL-6 and TNF-alpha. There is very little induction comparatively of interferon alpha with these small molecule agonists. And I just want to make a point here that in human clinical trials in cancer immunotherapy, multiple groups are using small molecule agonists similar to this that are not inducing high interferon alpha and are inducing high levels of inflammatory cytokines, which is not the profile that you want. So I just want to stress this. Activating that first site with small molecule agonist is inducing NF-kappa B, but not the IRF7 pathway. RNA agonists are not stronger than R848. They just switch activation away from the NF-kappa B pathway and towards the IRF7 pathway that can be measured by interferon alpha. So I think for the purpose of developing mRNA cancer vaccines, this could be a very important thing to keep in mind, that optimizing this may optimize the vaccine as well. Now, adding the data for a CpG-A DNA equivalent to vidutolimod, you can see actually that the level of interferon alpha is well below that of the RNA. This is not just any old published RNA. The published RNAs for activating TLR7 are actually no stronger than a CpG-A DNA at activating interferon production. This is a novel, improved RNA structure that we're using there. And when we combine that together with the TLR9 agonist DNA at a low concentration of 0.5, we see more than additive effects for improving the interferon alpha production. And that's without increasing the production of the inflammatory cytokines. So again, co-stimulation actually may be the way to go for inducing optimal interferon responses. Now, TLR7 and TLR8 are on the X chromosome. There are a number of publications that they may escape X inactivation in some immune cells, and there are some reports that females respond more strongly than males to TLR7/8 agonists. So we've started to look at that. This is just the data with the first two male and female responders, where if we look at the 7/8 agonist RNA versus the CpG DNA versus the combination of the two versus R848, we can see for each of these different readouts, there tends to be a higher response for the women compared to the men to the TLR7 agonist. The CpG DNA is about the same between the two sexes. So this requires more study, but it's an important thing to keep in mind if you're doing cancer immunotherapy or mRNA vaccines activating these sites, to be aware of the possibility that males and females may respond differently. In mice, we also saw evidence of TLR7/8/9 co-stimulation. This was data at 4 hours after injecting mice with either a standard MC3 LNP, similar to the LNPs that are in some approved human therapeutics, or a different LNP that actually turns out to induce much lower levels of IL-6 compared to the MC3, but much higher levels of CXCL10, indicating a qualitatively different immune response. And again, what's interesting with the 7/8/9, aside from the much higher induction of interferon alpha, and I just want to say, we have never seen serum levels of interferon alpha in mice anywhere close to that. In fact, usually we use CXCL10 induction as a measure for the type 1 interferon response because you just don't see a lot of interferon alpha in the serum. Something fundamentally different is going on with these agonists compared to agonists that have been published before. And then IL-12 induction is also certainly intriguing. We expect to have monkey data with these same agonists delivered IV in a few weeks, and very much looking forward to those to see what's happening there. So let me just give a high level of what we think is going on here. Pattern recognition receptors, this family of toll-like receptors, Rig-I-like receptors, Nod-like receptors, CGAS, STING, somehow these work together in a combinatorial fashion to detect danger and to classify that danger by whether it's infectious danger, and it could be extracellular infections or intracellular infections, or sterile tissue damage. It's really not clear exactly how the system does that, but I just want to propose a few points here. First of all, in terms of assaying for activation of those receptors, it's pretty easy to do at a high level. All of these receptors activate one or more of the IRFs, the NF-kappa-B, and the inflammasome pathways. So at a high level, if you just measure the downstream cytokines from those pathways, either in vitro assays or in vivo assays, you'll pick up if any of those systems are significantly activated. Reporter cell lines are fine for detecting activation of the NF-kappa-B pathway. They're really not as good in our experience for detecting IRF. None of the reporter cell lines seem to mimic the PDC very well. And in vivo assays in mice, you really need to look at 4 hours hours. 24 hours is too late to look for these early effects. In primates, 24 hours is fine. And then, of course, there's a lot of downstream effects and the generation of the adaptive immune response that follows the innate immune response, and that's been described elsewhere. I think as we consider these different threats, tissue damage occurring in the absence of infection, sterile tissue damage, leads to secretion of DAMPs, damage-associated molecular patterns. This is the state that tumors mimic. Tumors are releasing these DAMPs. They are mimicking damaged tissue. And there is some inflammatory effect of that. C-gas sting is activated. And in mice, that can lead to anti-tumor responses. In humans, it has not. These pathways ultimately lead to restoration of homeostasis with angiogenesis and tissue repair, and tumors depend on those pathways in order to protect themselves against immune responses. At the opposite extreme, the strongest signal to induce CD8 T cell responses has to be the retroviral infection, because once a retrovirus has infected a cell and integrating its DNA into the genome of the cell, the only way that infection can be cleared is to kill the infected cell. And this has to have been a very powerful evolutionary stimulus for CD8 T cells. Of course, other pathogens are also a threat, but coming back to the cells that we know regulate CD8 T cell activation, conventional dendritic cells that give signal 1 and signal 2 express TLR7 and or TLR8. Plasmacytoid dendritic cells that give that signal 3 express TLR7 or TLR9. And then the macrophages and the myeloid cells play a critical role. They can either support this CD8 T cell generation, or if they're coming from tumors or other inflamed, uninfected tissues, they can suppress CD8 T cell generation. So these cells have TLR7 and TLR8 as well. In the presence of a viral infection, these are activated by those receptors, and it appears that they become M1-like macrophages that can promote anti-tumor responses or anti-infective responses. Vidutolimod was only activating the PDC. It wasn't activating those other cell types. So it seems reasonable to hypothesize that co-stimulation of these receptors together may unlock a much more powerful CD8 T cell response than otherwise can be generated. So that should be the optimal way to go. And I think it's an important thing to consider in optimizing mRNA therapeutics. Again, for protein expression, you want to avoid the immune sensors. For infectious disease vaccines, you've got multiple ways to induce antibodies, including the lipids and the formulation, using double-stranded RNAs, or activating the TLRs if interferon alpha induction isn't an issue. For cancer vaccines, optimizing the CD8 T cell induction, I think, is going to require stimulating PDCs via TLR7 and or TLR9. And then keeping in mind the sex-based differences and not relying on mouse models to make decisions about what to advance into human clinical trials. Thanks, and I hope this has been helpful.